Hello, everybody, and welcome to today's 3P session. We are in our third week right now, and we are so happy that all of you guys are participating, signing up as speakers and as audience members. And by we, I mean the Cure Parkinson's Trust, the World Parkinson Coalition, and the Van Andel Institute. My name is Lisa Bergquist, and I'm a postdoc at the Van Andel Institute. And with me today, I have two fantastic speakers who are going to talk about some very interesting research. First out is Lynne Krohn, who's a PhD student at McGill, and she will talk about REM sleep disorder genetics for early detection and subtyping of Parkinson's disease. And if you guys have been with us earlier, you know the drill. Down at the bottom of your screen, you have the Q&A box where you can submit your questions throughout the session, and Lynne and Kariem will do their best answering them, and we'll try to get through as many as possible. So I'll just hand it over to you now, Lynne, and thank you.
Thank you. Okay. Sorry, I'm going to turn off my video. Okay. So yes, my name is Lynne Krohn. I'm a PhD student at McGill in the Gnor Lab for Neurogenetics. And today I'm going to be talking about REM Sleep Behavior Disorder genetics. Oops. Okay. So first I'm going to give an introduction on REM sleep behavior disorder, which I'm going to call RBD from now on for the sake of brevity. So what RBD is a lack of muscle atonia during REM sleep. So what that means is the normal paralysis that's supposed to occur in your muscles while you're sleeping to make sure that you don't move around is somehow disrupted. So patients tend to act out their dreams physically and or vocally. The prevalence is about 1% in the general population and about 2% in older populations, as the age of onset is commonly greater than 50 years. Much like Parkinson's, so far it's presenting as male dominant, and it's diagnosed by something called a polysomnography. So a patient will go into a sleep lab and be connected to a mechanism that records brain waves, oxygen levels, heart rate and breathing, and eye and leg movements. And because of this, because the necessity of staying over in a sleep lab and undergoing this test, we hypothesize that perhaps the prevalence is a bit greater than the estimated 1 to 2%. So the reason why I'm talking about RBD today is that over 80% of RBD cases will convert to a synucleinopathy within an average of 10 to 15 years. So synucleinopathy is characterized by aggregation of the protein alpha-synuclein, and most of RBD patients will convert to either Parkinson's disease or dementia with Lewy bodies, and in rare cases, sometimes multiple system atrophy. So what we have over here, it's a study by Ron Postuma, also at McGill, about 1,300 RBD patients. The I stands for idiopathic, meaning diagnosed by that polysomnography. And what we see is a survival plot. So at the 15th year, we have less than 20% of cases have not yet converted or dropped out of the study. So within within synucleinopathies, there are different levels of patients who have RBD and who don't have RBD. So within PD, about 30 to 50% of PD cases have RBD, and it's much higher in dementia with Lewy bodies. Here it's written DLB and MSA, which is multiple system atrophy. And a lot of the times in these subsets, they are diagnosed or identified by questionnaire instead of that polysomnography. So the questionnaires are highly accurate within synucleinopathies, so we're able to get these kinds of estimates. So like I said, I'll be talking about genetic studies of RBD, and there's several reasons that we are doing this. So one of our goals is early detection of synucleinopathies like Parkinson's disease. And for that, we can use something called a polygenic risk score, which I'll explain a bit in the next slide. We're also hoping to be able to predict conversion. So rapid converters from RBD to, for example, PD are going to be ideal candidates for clinical trials because it'll be a shorter trial time, less opportunity for dropout, less expensive, and just all around a better environment for a successful clinical trial. And also, finally, we're hoping to contribute to subtyping of Parkinson's disease. As a lot of you or everybody here probably knows, Parkinson's is sort of an umbrella term that encompasses a lot of different subsets. A lot of patients have different phenotypes, different rate of progression, different age at onset, and they can look very different from each other. So if we use genetics to try to help the effort of subtyping Parkinson's patients, perhaps down the line, more personalized treatments will be available. So these are our goals, and today I'm going to be talking about some studies that I have done so far in my PhD that have laid the groundwork, I think, for making these a possibility. So I'll be talking a lot about genome-wide association studies, which a lot of times I'll call GWAS, again, for the sake of brevity. And these have been done so far in each of the synucleinopathies that I mentioned that RBD converts to. And Parkinson's is the most widely studied because it has the largest cohort. So it's about 40,000 patients, over 1 million controls, and about 20,000 what they call proxy cases, all contributing to finding about 90 genetic loci that are associated with increased or decreased risk for Parkinson's disease. Dementia with Lewy bodies has a much smaller cohort. It's under 2,000, and multiple system atrophy even smaller. So for dementia with Lewy bodies, there's 3 significant genetic loci. For multiple system atrophy, there's none genome-wide significant yet. And today I'll be focusing on, for the most part, SNCA. So that's the gene that encodes for alpha-synuclein, so obviously plays an important role, likely in synucleinopathies like Parkinson's and dementia with Lewy bodies. So this is it's an example GWAS output, and it's just going to help illustrate what a polygenic risk score is. So you have, on the left there, we have a list of variants, and then you have the gene that the variant is closest to. And then because GWAS, it takes variants genome-wide, so from chromosome 1 to chromosome 22 usually, and does logistic regression, testing whether these variants are associated with increased or decreased risk of the disease you're studying. So what you get is a beta in that third column, which is a log transformation of the odds ratio. So what a polygenic risk score does is it sums the weight or the betas of each GWAS significant variant in each individual. So for example, if a person has variant 1 and what I called variant N down at the bottom, they would have a high risk score because if you look in the odds ratio, variant N gives you almost a 2 times more likely chance to have, for example, Parkinson's disease, and variant N actually has an 11 times greater chance to have Parkinson's disease. And that's actually a real variant for Parkinson's, well-known LRRK2 variant G2019S that highly increases your risk for PD. And on the contrary, a person with only variant 2, you see the second one, it has an odds ratio less than 1, so you're less likely to have Parkinson's disease, so you would have a low polygenic risk score. So what I did so far with polygenic risk score, of course, we're interested in the genetics of RBD, but we didn't know, now we do, whether RBD genetics are going to overlap completely with synucleinopathy genetics, considering the conversion. So what we did is took the GWAS variants for Parkinson's disease, created a polygenic risk score in a cohort of RBD patients and in PD patients and controls, and we did kind of a sensitivity analysis to see how well the polygenic risk score can differentiate between patients and controls. So what we see here, I don't think you guys can see my mouse, but on the right, we have PD versus controls. And you can see that there's a curve that represents, or that matches pretty well with this pink curve in the middle, this sort of comparison rock curve in the middle. And what that says is that it's a good curve. So that means it's differentiating fairly well between PD cases and controls. And again, this is a PD polygenic risk score, so that's a good sign for PD. But on the other hand, when we look in RBD, we have a curve that more closely resembles this blue curve, which the benchmark here calls worthless. So what that tells us is that the polygenic risk score for Parkinson's cannot differentiate between RBD patients and controls. So that means there's a possible distinct genetic profile of RBD that we are currently investigating now. So like I said, I'll be focusing on SNCA for the most part, and this is a study that we recently published a couple months ago, a fine mapping of SNCA in RBD and in overt synucleinopathies, which includes Parkinson's and dementia with Lewy bodies. So we had an RBD cohort of about 1,000 patients and 6,000 controls, and in a case control study, we found that there is a top risk variant for REM sleep behavior disorder in SNCA, and it's at the 5' end of the gene. So if you look at the bottom of these plots, they're called locus zoom plots. It's basically a focused Manhattan plot, but you can see very small the gene SNCA down in that box at the bottom. And you can see that the peak here, these are all individual variants and a log transform p-value. So you can see the peak for RBD here is at the 5' end of SNCA. There's also a secondary hit way out in the 3' on the panel on the right. So we did the same thing in Parkinson's disease, and this matches what they see in the larger genome-wide association studies, is that the top risk variant for Parkinson's is on the 3' end of SNCA. So the top risk variant for RBD is independent of the top risk variant for PD. So there's a different genetic profile sort of going on just in this particularly interesting locus for synucleinopathies. And actually the 5' risk variant for RBD is the same as the top SNCA risk variant for dementia with Lewy bodies. And this is potentially quite interesting because dementia with Lewy bodies, of course, is associated with a cognitive component of dementia. And Parkinson's patients with RBD tend to present with more cognitive issues and more PDD or Parkinson's with dementia. So there's a possibility that maybe this 5' variant is associated with some kind of cognitive component. And we're using, or with the help of collaborators, looking into that currently. So we kept investigating this particular locus, and what this is a beta-beta plot. So we took several variants in SNCA that are associated with various synucleinopathies, and we compared the betas from the RBD study to those from the Parkinson's disease genome-wide association study. And I'm just going to draw your attention to this variant here. This is the top risk variant for Parkinson's disease. And what this is telling us is that it's actually showing an opposite direction of effect in RBD than it does in PD. So while it's increasing risk for Parkinson's disease, it's decreasing risk. This is all association. This is not causal, just to make that clear. But it's associated with decreased risk for RBD, while it's associated with increased risk for Parkinson's disease. So that's pretty interesting. And what's more is that that 5' variant that was associated with RBD and dementia with Lewy bodies is actually more common in Parkinson's patients with RBD versus Parkinson's patients without RBD. So it's potentially an RBD specific locus. So again, just to sum that up real quick, there's a genetic difference between RBD and Parkinson's at a key genetic loci, that's SNCA. And also there is a genetic difference between Parkinson's with RBD and Parkinson's without RBD. So just to remind you, about 30 to 50% of Parkinson's patients have RBD, and that gets back to the aim that I mentioned in the beginning of this presentation, that this is evidence that we might be able to find genetic differences between the two groups on a larger scale and possibly create some kind of polygenic risk score to help differentiate between these two subtypes. So this is just to introduce some research that's happening now, but also to highlight the differences between Parkinson's and REM sleep behavior disorder genetics. So what this is another beta-beta plot, just like we saw in the last slide, except that these are preliminary results from a genome-wide association study that I'm doing on RBD. So now we're studying RBD genome-wide and comparing the betas to the 90 significant genetic loci for Parkinson's disease. So you can see the blue line here, or just the blue dots, those are variants that are in the same direction of effect, Parkinson's disease and RBD. So if they're both increasing risk, it's doing the same thing in both conditions. And then the red shows different directions of effect. And you can see there's quite a big difference for some of these. And something I will also draw your attention to is we see here at the bottom left is GBA. We have TMA 175 a little bit further up. And in the top right, KIRKAP2, INPP5F. Many of these genes are likely or definitely associated with the autophagy lysosomal pathway, which is very important has been implicated as a contributor to the pathogenicity of Parkinson's disease. So that's just one of the insights that we're getting studying RBD genome-wide, the sort of pathways that might be involved in RBD specifically. So one of the last things I'll touch on here, as I mentioned in my aims in the beginning, is that we're hoping to identify rapid converters from RBD to synucleinopathies for clinical trials mostly. And these are kind of proof of concept. These are very small studies at this point, but what we see here are survival plots. This is an SNCA variant. Sorry, I keep pointing, but you can't see me. The bottom, or the left panel, is an SNCA variant that is associated with more rapid conversion from RBD to any kind of synucleinopathy. And on the right, these are GBA variants. GBA is another very important gene in Parkinson's and dementia with Lewy bodies, likely multiple system atrophy as well. And what these are small studies so far that are giving us hope and giving us evidence that if we keep building these cohorts and refining this study, that we're going to be able to identify genetic markers for rapid conversion and genetically stratify patients for effective clinical trials. So I'm pretty much summing up. These are just a couple takeaways. RBD genetics do not completely overlap with known synucleinopathy loci. So this was evidence we needed to study RBD genome-wide, which is something I'm working on right now and hope to get the results to everybody within the year. And the second is the subtype of Parkinson's disease who have RBD may have distinct genetic markers. So again, this will be useful in genetically subtyping Parkinson's disease patients for hopefully more effective treatments. And what I'm doing here is another genome-wide association study, but this time with Parkinson's disease patients with RBD versus Parkinson's disease without RBD. So we're hoping to find genome-wide distinct genetic differences between the two groups. And finally, genetic markers may be associated with rate of conversion, and I've said a bunch of times this makes ideal candidates for clinical trials. We're continuing to build this study and collecting lots and lots of data to hopefully be able to more definitively show genetic markers that are associated with rate of conversion for use in this genetic stratification. With that, I'll say thank you very much to my lab, my supervisor, Dr. Ziv Ganor, all of our collaborators, and as always, for sure, all of the patients who make this research possible. And we're always looking for collaborators, or if you have any questions outside of this particular presentation, that's my email, my Twitter handle, and our lab's website. And also, if you're interested in learning more about REM sleep behavior disorder, these are two non-genetic reviews or studies by Birgit Hogel and Ron Postuma that I do highly recommend if this piques your interest. But for genetics, you can come to us.
Thank you so much for that very clear and interesting presentation, Lynn.
Thank you.
So we're going to move into the Q&A session, part of the session now. So I thought I was going to start asking you if you have, are there any variants that have been found that greatly increases the risk of RBD if you compare to similar what we see in Parkinson's with the G2019S variant?
So far, so for example, G2019S actually has not been associated at all with RBD. And actually Parkinson's patients with G2019S tend not to have RBD. So that particular variant, no. And so far, there's some variants in GBA that increase risk more than, like 1.2, 1.3 odds ratios, but we don't have a variant like G2019S yet in RBD, and we'll have to keep digging to see if we find something like that. But so far, it looks like no, there's not.
Okay. And we have a question here from Punet Chaudhary. Excuse my pronunciation. She wonders, and I think this refers to some of your background introduction slides. What are some of the other reasons for people to drop out of the studies besides developing PD?
It's you. Sorry, I'm just trying to find a nicer way to say, but if they pass away, they are dropping out of the study. But that particular study didn't have a high dropout rate for as in just discontinuing the study. So for the most part, that was just if they passed away. But those are fairly well-marked conversions.
Okay. And Elaine Dagger is wondering if there is a genetic correlation between PD and RBD.
Yeah, so that's something I'm working on now with the genome-wide association study for RBD, and I do have preliminary results, but our cohort, like I mentioned at the beginning with the dementia with Lewy bodies GWAS, we're about the same size so far. It's very hard to collect idiopathic RBD patients, like I mentioned, because of the polysomnography necessity and just that it's not that widely known as a condition. However, we have done genetic correlation studies, and there's not a definitive correlation between RBD and PD. We actually saw a negative correlation, but it's not significant, so that doesn't really tell us much, except that there's not a strong correlation there.
Okay. And you also in the beginning mentioned other synucleinopathies. Are you doing any of the same type of studies you explained here for RBD and other synucleinopathies?
Yes, dementia with Lewy bodies, yes, we are amassing a cohort to do a similar thing as far as comparing DLB with and without RBD. It's a bit different because so many DLB, that's dementia with Lewy bodies, patients do tend to have RBD, so there's not such a big difference. And also there's just a lot less samples to work with in dementia with Lewy bodies, but I did include a dementia with Lewy bodies cohort in that slide of the SNCA variant, and they were behaving the same way as Parkinson's. Actually, that risk variant that's associated with both RBD and dementia with Lewy bodies was actually showing up more in dementia with Lewy bodies patients with RBD versus without. So it's we are definitely continuing to try to build cohorts to do studies like this.
Yeah, because I was going to follow up and ask if you think you would see similar result for different synucleinopathies, or if it's going to be completely different results. But yeah, so
I think that they will be different because there's it's hard to compare dementia with Lewy bodies and Parkinson's at this time on the genetic level because the cohort for Parkinson's is just so much bigger, so they can find so many other variants. So it's hard to say like, oh, that variant's not associated in dementia with Lewy bodies, possibly just hasn't been found yet, depending on the odds ratio and how many people there are. Same with RBD. So, but yeah, we'll see.
So we have one question here from an anonymous attendee wondering if there's any type of treatment for RBD.
Yep. So they use a lot of times. I use melatonin. So they use melatonin, and that tends to help. Also, I'm going to bungle. I think it's clonazepam. But so they are just treating pretty much the movement and the actual phenotype of RBD.
Okay. And Susan Buff is wondering what the best way for a person with Parkinson with RBD to become part of a cohort, if not part of one already.
So we have the samples that I won't be the best person to explain this because I'm sort of on the receiving end of this, but I know that patients with REM sleep behavior disorder who are seeing a clinician on a regular basis, we tend to reach out to those clinicians and ask if patients want to share their data, things like that. But we also have something called the International REM Sleep Behavior Disorder Study Group. And this is a lot of clinicians from around the world that are contributing to studies like this. And I think that if a patient with RBD is seeing a doctor, they can bring, I don't know, actually, they could bring it up. But I think the International REM Sleep Behavior Disorder Study Group, there's a website for that that I can share, like tweet it or something. And there's also the NAPS Consortium, which is the North American Prodromal Syndromes Consortium, that's collecting RBD data and following up with patients longitudinally. So that's another good one.
Thank you. So Patrick Brandon is wondering if you think there's a difference in the distribution of pathology early stages of PD with RBD versus no RBD. So clarifying that, does RBD PD start in the brainstem while non-RBD PD would start maybe in the olfactory nuclei?
Yeah, it's there. I just saw a talk about this at McGill. This is being looked into, but it's not super definitive. But yeah, they do see like the RBD pathology starting in the olfactory and then the pons and things like that. So it's the one thing that I can say that is clear is that RBD patients, PD patients with RBD tend to present with like a more rapid disease progression and cognitive decline and things like that. So it's likely that there's going to be some kind of difference in the pathology, but that is not my expertise. So I'm going to stop before I say something that's wrong.
Thank you. And Neil Chatterjee is wondering if there are distinct, consistent pathological signatures for the synucleinopathy patients that presented with RBD, aligned with BRAC staging, higher level of RAF-LC path, etc.
Yeah, again, it's not my expertise, but they tend to follow the BRAC staging, similar to Parkinson's patients, and they do there have been studies of REM sleep behavior disorder patients post-mortem where they found alpha-synucleinopathy in the brain and in the earlier stages for the most part of the BRAC stages. So it's this synuclein is playing a role in RBD as well. That's what.
Yeah. And so Ariel Walker is wondering if there are any known genetic variants that seem to be protective from RBD conversion to synucleinopathies.
So far, not anything that I will speak definitively on. Yeah, I think I'll have to leave it there at that point. Our conversion studies are unfortunately small at this point, and I'm hesitant to say, even the ones that I showed, it's because they're so small, it's hopeful that we'll be able to grow our cohort and make these be more confident at saying that these are actually affecting rate of conversion. But so far that's in its infancy, I would say.
Okay, interesting. So we'll have to tune in later and hear about that.
Stay tuned.
So Miguel is wondering if restless leg syndrome is related to RBD because it also responds to L-DOPA.
Yeah, we are also doing a genetic correlation with RLS. I don't have anything to report on that at this point. And yeah, I think, yeah, that's about it.
Yeah, and maybe we can round out this session with the last question I have. So is RBD heritable? If my mom or dad or grandparents would have it, is it likely that I will have it as well?
Yeah, so there are studies that have shown that you are more likely to have RBD if you have a family member who has RBD. It's not a huge increase. Like it's not like if a parent has RBD, you're 10 times more likely. I believe it was much smaller than that, but it's also something that I'm calculating with the genome-wide association study is we can calculate the genetic heritability of RBD, and in a smaller cohort, we found it was very similar to Parkinson's disease. So that's about like 25%. And now I'll be doing that in the larger cohort. But the bottom line is it appears to be somewhat heritable, yes, but not super high. Again, like we were saying with the variants that are increasing your risk very much. We haven't found those in Parkinson's disease, I mean in REM sleep behavior disorder. And that's what would make it, super highly heritable. So we will see, but the answer is yes, to some degree.
Excellent. Thank you, Lynn, so much for that great presentation and for answering all of our questions.
Thank you.
So we're going to move on to our second speaker today, Kariem Ezzat, who's a research scientist at Karolinska Institutet in Sweden, and he will be talking about loss of function mechanisms in amyloid pathology. So please take over the screen, Kariem.
Thank you very much, Lisa, and the organizers for the opportunity to talk here. I'll try to share my screen without messing this up. Just a second. Sorry. Share screen. Okay, so now is it? You see my screen? Okay, yes. So I'll be talking today about a different topic related to the biophysics of amyloid. So I'll be talking about a mechanism of amyloid aggregation called the heterogeneous nucleation mechanism, and how is it related to postulated loss of function of amyloid as a mechanism of amyloid pathology. So first, just a brief introduction. Of course, everybody here is familiar with amyloids, that they are fibrillar protein aggregates that are associated with many diseases like Alzheimer's disease, Parkinson's disease, Huntington's disease, and prion pathologies. So they are kind of widespread disease pathogenic mechanism. So the first question is, what are amyloids? And it's important to highlight that amyloids are not particular proteins. They are a state of proteins. And as you see in this video, it's like as if it's two different states of the same thing. So you have water that starts to nucleate and then transforms into this very beautiful snowflake. So it's a transformation from a soluble or liquid state to a solid state. And this mechanism of phase transformation, as we will discuss later, is very similar to what happens when proteins transform from being soluble in their native form to being to transform into solid amyloid fibrils. So the amyloid state, as a physical state of proteins, has various traits. First trait is like it's universal. There are nearly 40 proteins that can form amyloids in various human diseases, and these proteins vary from p53 to insulin to amylin, and of course the famous ones like amyloid beta in Alzheimer's disease, alpha-synuclein. But it's a very universal phenomenon. It can happen to many proteins in the organism, and they can cause many diseases other than the ones that are famous, like the neurodegenerative disease. For example, amyloids of p53 can cause cancer. Amyloid of diabetes can cause amyloid of amylin, which is islet amyloid polypeptide, can cause diabetes. So it's a universal phenomenon that can happen to basically any protein. It's also a generic phenomenon. So like all amyloids share the core molecular conformation, which is the cross beta conformation, which are two beta sheets of the peptide facing each other as what is called a steric zipper. And this gives the amyloids their distinctive fibrillar structures, where you have these beta sheets with the peptides stacking over on top of each other, and then they the beta sheets face each other in the cross beta conformation, forming these fibrils. Another important trait or characteristic of amyloid is that amyloids are very stable. Many scientists believe that the amyloid fibrils are at the bottom of the energy landscape of proteins. So proteins, when they fold into their native state should be stable, but not very stable, because it needs to be reversible. However, when it reaches the amyloid state, it's so stable that it becomes irreversible. So the process of amyloid transformation. So just to wrap up, these are like some of the general phenomena, that amyloids are a state of proteins. They are not a particular protein. They are universal, can happen to many proteins, and it's a generic protein conformation. They share the similar core structure, and they are very stable. So the process of a protein transforming from being normal and soluble to being an amyloid involves three pathological transformations. First, the protein transforms from natively folded to the cross beta conformation. This is a biochemical transformation. And second is the biophysical transformation, from being soluble to being insoluble. And third, a biological transformation from being functional to being non-functional. So the biochemical transformation you see here, a natively folded protein, then the protein under certain circumstances start to adopt this cross beta sheet architecture, and then keeps on in the elongation to form these fibrils. But the core conformation is the cross beta conformation. Another important transformation is the transformation from being soluble to being insoluble. As you see here in this example, I will just stop for a little bit. So in this example, we have supersaturated solution of a salt, and it's going to crystallize. And the process of crystallization is very similar to the process of amyloid transformation. We have a protein that is soluble and function, and somehow something pushes it above a certain barrier, transforming this soluble protein from the soluble state to the insoluble state or to the crystalline state in this case. So you see here, this is a supersaturated solution. However, when you add an exogenous surface like spherical metal beads, these act as catalytic surfaces that kick the reaction of transforming this soluble salt into forming these elongated crystals. And the same reaction can be kicked off by adding a preformed, nucleus or preformed crystal. And this little demonstration shows the two of the pathways of how the phase transformation of amyloid from soluble to insoluble can occur. It can occur either via two catalytic mechanisms: seeded nucleation, when you add a preformed seed or preformed amyloid or a prion, it drives or triggers the transformation directly, or you add an exogenous surface, like in the first case it's called seeded nucleation, second case is called heterogeneous nucleation, as shown here. And these are the differences between the two types of nucleation: the homogeneous nucleation, when this the soluble proteins or the soluble salts form this spontaneously form this nucleus, and this nucleus kicks off the phase transformation reaction. It's called homogeneous nucleation. However, when you have exogenous surfaces shown here, like for example, like the vessel wall, these surfaces can also kick off the nucleation reaction by lowering the energy barrier, increasing the local concentration of the protein, and inducing conformational changes. So I'll not talk a lot about the nucleation barrier, but you have this free energy barrier, or that a nucleus needs to be formed for the phase transformation to happen. And this is what we showed here, that without adding this preformed nucleus, nothing is happening until you add a catalyst. And this catalyst can be this exogenous surface, and it's, in this case, it's called heterogeneous nucleation, or it can be a preformed seed, and it's called seeded nucleation. So without catalysis, this system would remain metastable and soluble. And this is very important in terms of understanding how amyloids form, because when you study amyloids from a biophysical point of view, this is the type of curves you get. It's like amyloids, if amyloid proteins that form amyloids, like amyloidogenic proteins, can also form amyloids via two different pathways. Either if at very high concentrations, then nucleation can happen spontaneously, but then there will be this lag phase for this nucleus to form spontaneously. And then once it's formed, the elongation reaction takes place immediately. However, if you do a catalyzed nucleation, if you add a preformed seed or a heterogeneous surface, then you can bypass this nucleation phase, and the amyloids are formed immediately, bypassing the nucleation phase. I know for many of the listeners that are not familiar with these terms, this sometimes sounds confusing, but it will be relevant to the pathology in the next slides now. So, for example, the nucleation reactions are not only happening for the amyloids; they also happen for many other proteins. Like here, for example, actin. So actin is a fibrillar protein, and for it to form fibrils, it also needs to go through the nucleation and elongation reaction. But in case of functional proteins like actin, in this case, this nucleation reaction is very, very well controlled by highly evolved nucleation proteins and energy-dependent mechanisms, and it's a very reversible process. So in this video, you see like this protein is called spire, and it acts as a heterogeneous nucleation surface as a catalyst for actin to form the fibrils. So you see this, and this is a heterogeneous nucleation, but this heterogeneous nucleation happens for a functional protein, a functional fibril. And you see in this case, the G actin, the actin like subunits form this fibril, but they form it in their native conformation, not in the cross beta conformation. So this is a functional protein that forms fibrils, but they form functional fibrils in its native conformation in a very, very well-controlled nucleation process. Without this nucleation catalyst, which is this protein called spire, actin is prevented from forming these fibrils by special machinery, special proteins, special mechanisms that prevent actin from spontaneously forming fibrils in an uncontrolled manner. So you have all these proteins that bind to the actin monomers like profilin, tyrosine, and capping proteins that prevent actin from just spontaneously forming a nucleus, because this nucleus, like with homogeneous nucleation, can occur, and then actin fibrillization will happen uncontrollably. So this is, here we see how biology evolved to control nucleation mechanisms to form functional fibrils. It has to be a very well-controlled mechanism, and it has mechanism to start it, and mechanism to inhibit it, and mechanism to reverse it if it wants to unfibrilize the actin. The same happens with collagen, for example. So the same mechanisms happen in collagen fibers. They also grow by nucleation, but also they are formed of subunits that are not cross-beta subunits like the amyloids. They are formed of subunits that are helical structures that are rich in prolines and glycines. And also here we see that there are evolutionary barriers to prevent the functional fibril structures from going into the amyloids, which are uncontrolled fibrillization. In this case, these collagen subunits are highly rich in proline and glycines, which are amino acids that prevent the formation of beta sheets, so that the structure doesn't go into the cross-beta conformation. So in that sense, in the normal fibrillization reactions, they are controlled by nucleation. They are very, very well-defined mechanisms, and they are rich in amino acids that prevent the cross-beta conformation. Unlike the pathological nucleation, which is uncontrolled, so proteins are forced to polymerize, proteins that are not usually need to polymerize, like insulin or prolactin or all these proteins, or P53. They don't need to polymerize into fibrils to function. They function in a different form, but they are forced to fibrilize with a very uncontrolled mechanism. It's an energy independent manner. It forms beta sheets, so the proteins have to unfold and denature to form these fibrils, and it's irreversible. So there are physiological polymerization and there are pathological polymerization. The amyloids are in the category of pathological polymerization. 40 proteins that are very functional in many and have a lot of functions in the organism that are forced to polymerize and form non-functional polymers and lose their function. So just to keep the time. So one way that has been found in maybe 13 years ago now, that nanoparticles can force certain proteins to form fibrils, to form amyloid fibrils. So these nanoparticles can act as heterogeneous nucleation catalysts that can induce the nucleation of particular proteins. In this case, it was myoglobulin. And this is the curve of the amyloid nucleation. When you have the nanoparticles, it accelerates the process of amyloid aggregation by acting as catalyst for nucleation. So what we did in a paper that we published last year is to consider viruses as nanoparticles. So viruses are obligate intracellular parasites, and they are nanosized. So outside the cell, they are biophysically equivalent to nanoparticles. So we asked the questions whether viruses can also act as catalytic surfaces that force certain proteins to transform from being soluble and functional to being insoluble, polymerized, and non-functional by acting as catalytic surfaces via heterogeneous nucleation. And we chose herpes simplex virus because the herpes simplex virus has been shown to have associations with Alzheimer's disease. So here in this case, you see that the people who have activated herpes simplex virus are more likely to get Alzheimer's disease. So we found that when we incubate the herpes simplex virus, with amyloid beta peptide 42, the herpes simplex virus does what the nanoparticles do. They act as a catalytic surface. It binds the amyloid beta 42 peptide, concentrates it, induces conformational changes, and leads to the accelerated amyloid aggregation. So in this sense, we're trying to find a direct mechanistic link between viruses and amyloid pathologies. And this direct mechanistic link is based on the heterogeneous nucleation, where viruses act as catalytic surfaces for bypassing the energy barrier required for amyloid formation. And here we show that the virus interacts directly with amyloid fibrils. And when we did this experiment in animal models, where we injected the virus in 5X FAD models, we see that the virus catalyzes amyloid aggregation and accelerates the process of amyloid formation. 48 hours after infection, we get so much enhanced amyloid aggregation in the brains of these mice. So based on this, on these concepts, we view the process of amyloid aggregation as a process of pathological protein polymerization. You have a protein that has a certain function that is protected from being polymerized by the nucleation barrier, by the chaperones, by the proteasome, by all the mechanisms that keeps the protein in its native conformation to be functional, but somehow it's forced to polymerize, it's forced to cluster, to aggregate by certain cofactors or catalysts. That these cofactors can be endogenous or exogenous. And then once it forms these polymers, it stops being functional. And this, of course, can contribute to the pathology like milk. When it curdles, if you have curdled milk, it's of no use putting it on coffee because, I mean, it's lost its solubility. So based on this mechanism, we try to develop this scheme of trying to understand amyloid pathologies based or the etiology of amyloid pathology is based on the mechanism of nucleation, where hereditary amyloid pathologies are more dependent on spontaneous nucleation, homogeneous nucleation, where overexpression can facilitate the nucleation, so spontaneous nucleation leading to aggregation of amyloid fibrils, or that structural mutations that structural mutations that can lead also to facilitation of the formation of the nucleus that can also lead to amyloid aggregation. However, this can explain a very small proportion of the amyloid pathology, because hereditary forms are maybe maximum 5% of all the cases. So for the sporadic forms, we hypothesize that they are probably more dependent on other mechanisms that are dependent more on surfaces, on heterogeneous nucleation mechanism. Of course, there is a part of the sporadic that is dependent on seed-based nucleation, where you have prions that can help propagate the pathology between organisms or within the organism. But we think one of the most ignored mechanisms of inducing amyloid aggregation is the reaction on surfaces via heterogeneous nucleation. There are so many surfaces that can act as catalysts for the induction of amyloid aggregation by catalyzing the nucleation step. And these surfaces can be of microbial origin, like viruses, bacteria, fungi, or protozoa, or of non-microbial origin, but pathological surfaces still, like lipid pathologies, pollutants, nanoparticle pollutants, even microparticles that are released from like traumatic brain injury. Can act as catalytic surfaces for induction of amyloid aggregation. So within this scheme, the proteins are forced to polymerize, to change the conformation, that under certain conditions we are subjected to catalysts that are otherwise absent in their native microenvironment. And these catalysts, especially these pathological surfaces, will lead to this catalyzed nucleation and induction of conformational changes that will lead to the loss of function of the protein. But this leaves us with a dilemma that how about the protein-only hypothesis? How about the protein self-replication? Where does it fit in this scheme? Unfortunately, it doesn't fit in this scheme, because within this scheme, proteins cannot store or transmit biological information. It's just a process of crystallization. As you've seen in the beginning of this presentation, there is no information transferred from these beads to the growing crystal, and there is no information transferred from the surface to to the proteins, because surfaces can induce amyloid aggregation, and they are not just cofactors, they are independent factors that can induce amyloid aggregation in absence of seeds. So you don't need a seed, or you don't need a prime to induce induce the amyloid formation. You can do this just with a surface, you can do this with a nanoparticle, you can do this with a virus, independently of any seed or any prime. So primes are just one way of reaching the amyloid state, but there are many other ways that do not involve prime pathologies. And in this pathways, especially in heterogeneous nucleation, where you have surfaces, there is no information transferred from the catalyzing surface to the growing fibril. And in that sense, it's just a process of phase transformation similar to crystallization that has nothing to do with transformation of biological information. And in that sense, the different polymorphs that are formed by amyloid cannot actually be faithfully replicated, because neither the information can be stored in the cross beta conformation, and they cannot be transferred because they are dependent on factors that are not encoded by the structure. Since I'm running out of time, I will not go through this table comparing the cross beta sheets to the double helix of the DNA, which is the actual biological polymer that transfers biological information. But hopefully our paper that was just accepted in Frontiers Neuroscience a couple of weeks ago will appear soon, and it has all the information. I will share it on Twitter when it comes out online. So just to sum up this part, that amyloids are proteins behaving like proteins. They are not proteins behaving like viruses. They can polymerize, solidify, precipitate, change conformation, have a function, and lose their function. And these mechanisms happen to actin or collagen, but they are in a normal context. They are very well controlled. But when they happen to proteins that are not supposed to polymerize, that are not supposed to form fibrils, these proteins lose their function. And they are forced to do this by exogenous factors that involve microbes. So, and this gets us to the loss of function. Quickly, what is the evidence of loss of function in amyloids? There are a lot of animal models where when you knock out the proteins that form amyloids, you get a phenotype. And we have this box also in the paper. I'll not go through it because I'm running out of time. But for example, for alpha-synuclein, for example, there are several papers showing that mice lacking alpha-synuclein display functional deficits. The triple knockouts mice have age-dependent neuronal dysfunction, and when you do, in vivo RNAi knockout of the alpha-synuclein in adult mice, you also get Parkinson's phenotype when you deplete these mice from alpha-synuclein. So it seems that the alpha-synuclein in this case, losing the alpha-synuclein is a pathogenic mechanism. And we hypothesize that when alpha-synuclein aggregates into amyloid form, it loses its function, and it leads to these type of phenotypes based on the loss of function. I'll not go through this, but just quickly that the soluble proteins like amyloid beta 42 in the CSF of Alzheimer's patients, or soluble alpha-synuclein Parkinson's, they go down by age and they don't go up, which also might indicate that the loss of function of the soluble fraction of alpha-synuclein and amyloid beta is contributing to the pathology as well. So an example of loss of function is insulin-derived amyloidosis, where these patients, when they inject insulin in the same place every time, these are diabetic patients, insulin form amyloids under the skin, as seen here in this picture. However, these amyloids are like kind of benign. They don't do anything to the patient, but the patients still suffer from poor glycemic control, not because of the amyloids that are formed, but because they are not benefiting from the insulin that they are taking. So the solution to these patients is actually changing the injection site so that to give them more functional insulin, which restores their glycemic control. So this is an example of an amyloid pathology that is sorted out by replacement therapy and not by targeting the amyloid itself. So this is the model that we propose, is that you have the soluble fraction is going down over time by being transformed into an aggregated form. But when the soluble form reaches down to a certain level below a certain threshold, you get a pathology, and then after a certain time, when the aggregation reaches very, very high levels, maybe you start to getting a gain of toxic function, toxicity. However, in the early stages of the disease, probably the loss of function is very critical for the pathology compared to gain of toxic function mechanisms. And how this is related to therapy, we have tried everything in Parkinson's and Alzheimer except replacement therapy, except trying to restore the function of these proteins that are not functional. So this is just a schematic of all the hypotheses that have been tried in clinical trials. So what are the loss of function therapeutic strategies? If we consider loss of function as contributing to the pathology, there are two ways of going about this. First, to stabilize the soluble protein, or to replace this protein with a non-aggregating analog. So stabilization of the soluble protein, and these are the two only approved drugs that deal with amyloid pathologies, and they are both within the loss of function paradigm. So you have tafamidis, which stabilizes the trans-13 soluble form, prevents it from forming aggregates, or you have what's it called, pramliteid, which is a non-aggregating form of the amylin, and here's its structure. This human amylin forms amyloids in diabetes mellitus, so it's a replacement therapy. When you give pramliteid, which has these modifications, proline modifications to prevent the formation of cross-beta structure, then it's a non-aggregating replacement therapy. So this is my last slide. The integrated therapeutic approach from this paradigm, the heterogeneous nucleation loss of function is twofold. First, to target the pathological nucleators, especially like the surfaces that cause amyloid aggregation, like microbes and lipid pathologies, and also replace with non-aggregated analogs. And thank you so much. Sorry, I couldn't squeeze it in less than that, Lisa. I'm very sorry.
No worries.
Yeah.
Thank you. That was a very interesting presentation, Kariem.
Thank you.
So I'm just going to start asking you some questions so we can see how many we'll get to squeeze in before we need to round this up. But again, thank you for that very interesting presentation. So in your slide where you had the seeding and then heterogeneous nucleation in sporadic disease, like what do you think is the main driver of amyloid formation in these types of diseases?
I think in the sporadic sense, you don't have a genetic mutation that will either cause an overexpression of the protein, which leads to crossing of the nucleation barrier, because when you have high concentration, it's easy to form this nuclei, and then it causes amyloid aggregation. Or you have genetic mutation that makes the protein structurally more prone to self-assemble and form these nuclei. So if in the absence of this mutation, the only mechanism that we can think of that is causing amyloid aggregation is exogenous, is pathological surfaces, heterogeneous nucleation surfaces. That's something that is coming out in the microenvironment that is not supposed to be there, and it starts binding these proteins and inducing their conformational changes and catalyzing the amyloid aggregation. That's why I think it's for the sporadic forms, maybe the surfaces, the heterogeneous nucleation surfaces are etiologically more important in that sense. And this is what we try to show with viruses, for example, that when the viruses, we inject the viruses, it accelerates the process of amyloid aggregation. And this can be extended to other pathologies as well.
Yeah. So Sarah Elfarash is asking in regards to replacement therapy. Do you mean adding more alpha-synuclein? Don't you think this will exaggerate the aggregates?
Yes, a very good question. But that's why when we say replacement with a non-aggregating form. It's like pramliteid, this amylin analog. So it's a replacement therapy for a protein that forms amyloids, but it's chemically modified by changing some amino acids to prevent the formation of the beta sheets, so that it has the same function when it's soluble, but it cannot form the aggregates. So, and this is what we're trying to design now, to design alpha with collaborators here in Sweden. We're trying to design synuclein analogs that have the same soluble structure, but cannot form these beta sheets.
Okay.
Yeah.
Thank you so much. Unfortunately,
Thank
we're
you.
out of time now. Please continue discussions on social media if you have the opportunity to do that. I see there's a lot of questions pouring in now, and it was a very interesting talk. And I'm just going to ask Mickey if you want to tell us about who will be joining you on our next session on Thursday.
Thank you. Thank you, everybody, for speaking today, and thanks, Lisa, for chairing. So this Thursday on the 16th of April, we'll have another 2 speakers at the same time. We have Sarah, a postdoc from the NIA, presenting on genetic analysis of ALS, identifies contributing pathways and cell types, and Nicole, a postdoc at Johns Hopkins, who will be presenting on Parkinson limits priming and activation of the NLRP3 inflammasome in dopaminergic neurons. So if you're interested in these topics, please tune in on Thursday. And if you're interested in presenting, please email the 3pseminars@gmail.com. Thank you, everybody.
Thank you. Thanks. Bye.
Bye.
Thank you all.